Expression of proteins in prokaryotes. Creative Biomart. Principle and protocol. Introduction. A method in which a cloned gene is inserted into a suitable vector and introduced into Escherichia coli for expression of a large amount of protein is generally referred to as prokaryotic expression. This method has applications in protein purification, localization and functional analysis. E. coli used to express recombinant proteins has the following characteristics: easy to grow and control. Materials used for bacterial culture are less expensive than mammalian cell systems. A wide variety of E. coli strains and matching plasmids with various properties are available. However, proteins expressed in E. coli often form inclusion buddies to affect the biological activity and conformation of the expressed protein due to lack of modification and post-translational processing such as glycosylation and phosphorylation. Creative Biomart. Introduction. Expression vectors play an important role in genetic engineering. Prokaryotic expression vectors are usually plasmids. Typical expression vectors should have the following components. 1. Selecting a coding sequence of the marker. 2. A promoter capable of transcribed. 3. Transcriptional regulatory sequences, transcriptional terminator, ribosome binding site. 4. A multi-restriction cleavage site linker. 5. Sequences autonomously replicated in the host. Creative Biomart. Introduction. The general procedure for prokaryotic expression is as follows: obtaining the gene of interest, preparing the expression vector, inserting the gene of interest into the expression vector, sequencing verification, transforming the expression host strain, inducing expression of the target protein, analysis of the expressed protein, amplification, purification, further detection. Creative Biomart. Primary reagent: 1 pounds medium. 2 100 ml isopropyl thiobeta-D-galactoside. 2.38 grams of it was dissolved in 100 milliliters of DDH2O, filtered through a 0.22 μm filter, and stored at -20 degrees Celsius. Creative Biomart. Experimental procedure: 1. Obtaining the gene of interest. 2. Construction of recombinant expression vector. 3. Obtaining the expression strain containing the recombinant expression plasmid. 4. Induced expression. Creative Biomart. Precautions. 1. When selecting an expression vector, it should be considered according to the final application of the expressed protein. For ease of purification, fusion expression can be selected. If a native protein is obtained, non-fusion expression can be selected. 2. In the fusion expression, when the foreign DNA is selected and linked to the carrier molecule, the reading of the cryptographic structure during transcription and translation cannot be interfered. Collected by Creative Biomart. Creative Biomart. Contact information. Email: contact@creative-biomart.com. Website: www.creativebiomart.net. Address: 45-1 Ramsey Road, Shirley, New York 11967, USA. Contact us.